Let's switch gears now and talk about blood types. This is usually done using the ABO format, and maybe you've given blood or hopefully not had to receive blood, but maybe you know your blood type. But we refer to our blood types as type A, B, AB, or O. So when we do that, we're talking about the presence or the absence of specific antigens, and they're located on the surface of red blood cells. Now, when we talk about something being positive or negative, we're referring to another antigen called the Rh antigen. It was discovered later. That's why it's not included in the ABO, and we call it positive or negative. Now, these are called antigens, and they're glycoproteins, so proteins with a little bit of sugar on there, and they're on the surface of the cell. All right. They may also be known as agglutinogens. Now, when we do this actual exercise in the lab, what you'll see is agglutination or clumping. So more on that in a little bit. Okay, so the type here is the type of antigens that are present. Okay, so that's really important. So type A blood has A antigens on its surface. Type B blood has B antigens on the surface. Type AB has both A and B. And type O, it's a letter O, but it really means 0 antigens, so it doesn't have A or B. And if it has, so Rh positive means it has the Rh antigen. Okay, so let's do an exercise here. What specific antigens would be present on somebody who has type A positive blood? Not A plus, A positive. Okay, so that's going to have type A blood has A antigens, and positive blood means it has Rh antigens. Now, let's go ahead and talk about antibodies. And more will be covered on this in the lecture, but be patient. But here's your brief little intro. Antibodies are Y-shaped proteins, so they kind of look like this. And they're made by B lymphocytes. And each particular B lymphocyte only makes one type of antibody. And when they make them, they release it into the bloodstream. And what those antibodies do is they just go through the bloodstream, and they are looking for the antigen that they are bound, that they fit with. So they're trying to find it. So the antibody has a very specific shape on the ends of the Y, and that's going to attach to whatever specific antigen. Here we're talking about blood, but it can be a parasite, a virus, a bacteria. There. All right, so when it finds it, it attaches. And when it attaches, it marks the cell for destruction. So the way that this appears in the blood is this process called agglutination. So it causes these cells to clump together. So agglutination means they're going to clump together. Now, blood that's clumped together can't flow, and so that's very bad. You do not want this to happen inside the body. That's why blood types are really important. So that's why we get the term, the antibodies cause agglutination. So these antibodies are called agglutinins. So agglutinins plus agglutinogens, up here, agglutinogens, cause agglutination. That's kind of confusing, so you might want to write those out and those definitions a couple of times when you're studying. So now, it's really bad for somebody to react to their own blood. We don't want that to happen. So the way the body does this, somebody that has type A blood, meaning they have A antigens on their blood cells, they are only going to make anti-B antibodies. So if the person made anti-A antibodies, we would have the formula for antigen plus antibody equals destruction. And so this person would be destroying their own blood. So they would say their own blood, we're just trying to use all the words, agglutinates. So that's going to be somebody that's not going to end well. Okay, so we do this exercise in the lab, and we're going to, in the medical lab as well. So what you do is you can test what's on a red blood cell. So you'll take a sample and combine it with different types of anti-serum. So anti-sera is the plural there. These are solutions that contain the antibodies that are going to bind to this stuff. So if antibodies against A, that would be called anti-A, are mixed with a sample of the blood, it's going to clump. So you're going to get this agglutination. And so this clumping, when that happens, so you get this visible clump. It's very obvious. You will see it. So this is called agglutination. We're just writing the words again. Okay, so here is just like a sample. This is somebody and they have A antigens. So when we take this blood and we have our A antigens mixed with our anti-A, so anti-serum, so the anti-A antiserum, you get this clumping. This does not look like blood. That's bad. We do not want this to happen in the body. So that's why we can't, we have to give somebody the correct blood type. Okay, so anti-A serum, and this is easily confused, so you're gonna want to go over this several times, because this portion here talking about blood typing is not covered in lecture. So make sure you go over it, go over the handout, go over the lectures, and make sure you understand this. So anti-A serum has anti-A antibodies. They're going to react with any blood that has an A antigen. All right, the pattern continues. Anti-B serum has anti-B antibodies, and it's going to react with any blood that has a B antigen on the surface. So remember, reacts with. So reacts with, that means clumps, forms like agglutination, antibody plus antigen. And then you also have anti-RH, has anti-RH antibodies, the serum, and that's going to react with any blood that has the RH antigens. That's where you get the positive or the negative.